Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall look at some of the biochemistry that is associated with the digestion of lipids as well as its absorption. So just a reminder, we are still talking about the postprandial phase, what happens to your lipids after a meal. In the previous video, we talked about how glucose can provide us with ATP by going through what we called main street. In the process of main street, we can see a lot of intermediates that are formed. So one of these intermediates is acetyl-CoA. So in video 5.13, we saw how we can take this acetyl-CoA and make fatty acids out of the acetyl-CoA. And then in video 5.14, we saw how these fatty acids can be subsequently converted into triglycerides. We then discussed the synthesis of amphiphilic lipids such as phospholipids and sphingolipids in video 5.15, and then finally in video 5.16 we talked about how we can take this acetyl-CoA and make cholesterol. Since a lot of this synthesis, lipid synthesis, requires NADPH, in the next video, in video 5.17, or in the last video, we talked about the HMS, a hexose monophosphate shunt pathway, or the pentose phosphate pathway, and this is the pathway where we get most of the NADPH that is required, especially for the synthesis of lipids. So in today's video, we'll look at what happens to the lipids that you ingested in your diets. Okay, so first we are going to talk about, there are two parts. So first we're going to talk about the digestion part. Okay, we basically digest these lipids into chemicals that we can absorb into our bloodstream. So the food enters the mouth and then eventually the stomach, and let's look at lipid digestion in the mouth and the stomach. The lipids in the food will, in the presence of water, form these large lipid droplets, and that's how the lipids will exist in your mouth as well as your stomach. Those lipids that are very water insoluble, that don't have any part that likes water, such as cholesterol esters or the triacylglycerol, also called triglycerides, they will be buried more in the droplet. They'll be inside the droplet. However, those lipids that have parts that like water, such as in phospholipids, you have the head that likes water. In cholesterol, you have the hydroxyl group that likes water. In sphingomyelin, once again, you have the head that likes water. These lipids will be more towards the surface. Okay, so some lipids will be more on the surface, other more hydrophobic lipids will be deeper, buried inside the droplet. And your water molecules will be outside, facing the surface of the droplet. So if you look at the lipids that you find in your food, for example, the triacylglycerols, you remember that triacylglycerols have fatty acids. You can see the fatty acids here. Your phospholipids have fatty acids. Sphingomyelin has fatty acids. Cholesterol ester has fatty acids. And these fatty acids can be a variety of different fatty acids. For example, you can have the short-chain fatty acids, roughly about less than 8 carbons. You can have the medium-chain fatty acids. You can have long-chain fatty acids. And you can have very long-chain fatty acids. So each one of these is a class of molecules where the fatty acid is different for one C compared to another C and so on. And then in these steroid classes, in your diet, you could have free cholesterol, you could have cholesterol ester, you could have sterols coming from plants. These are called phytosterols. And of course, you have a variety of phospholipids. The diet gives you about 2 grams phospholipids. Bile that comes into your digestive system later in the small intestine, that has a lot of phospholipids. And then your diet has a small amount of sphingolipids, such as sphingomyelin. So that's basically how the lipid droplet will exist in your mouth and your stomach. Now there is an enzyme in your stomach called gastric lipase. There's a small amount of lipase in your mouth, and that's called lingual lipase. And these lipase enzymes will break down triacylglycerol, triglycerides, especially at position number 3, and give you diacylglycerol and free fatty acid. So if this is a triacylglycerol or triglyceride, you would have a break at the last carbon here, and you would have a fatty acid released, this one here, and then the rest of this molecule will be diacylglycerol. So notice that this product here, diacylglycerol, so we have a polar group here, and then we have the 2 tails. So this really becomes will have some surfactant properties, just like what we see here with phospholipids and what we see here with sphingomyelin. So as more and more of this surfactant diacylglycerol is formed due to the action of the lipase enzymes, so this surfactant concentration will increase, and together with the movement of the food due to peristalsis, this droplet will be converted into smaller droplets, typically less than 500 nanometers, and these finer droplets will then enter the small intestine from the stomach. So now the food is in the duodenum. That's the first part of the small intestine. So the duodenum gets bile, and this bile is made in the liver but stored in the gallbladder. And the duodenum also gets pancreatic juice that comes from the pancreas. Bile is very rich in bile salts. These are detergents or surfactants, and we'll see these play a very important role in digestion in the small intestine. While pancreatic juice is rich in bicarbonate, that gives it a more alkaline pH, as well as several digestive enzymes, very powerful digestive enzymes. So in the small intestine, the bile salts will bind to the lipid droplet, and together with that there is a small protein called colipase that will also bind to the lipid droplet. Now colipase, the small protein, stabilizes the active structure of lipase and also increases the hydrophobic binding of lipase to the oil droplet. So in the presence of colipase, the enzyme pancreatic lipase will now bind to the lipid droplet. So now we have this pancreatic lipase on the surface of the lipid droplet. Now pancreatic lipase will now go and start hydrolyzing the triglycerides, start breaking down the triglycerides in the lipid droplets. But they can only do that in the presence of co-lipase. So if we did not have co-lipase, then the triglycerides will not be broken down and there will be fat in the feces, and that's called steatorrhea. So once we have the pancreatic lipase, the bile salts, and the co-lipase, now we are ready to hydrolyze or to break down the triacylglycerols that are present in the lipid droplet. The pancreatic lipase prefers position 1 and 3 of the triglyceride. So right here, position 1 and position 3. And that will give us diacylglycerol and fatty acid, as well as monoacylglycerol and fatty acids. Another enzyme called carboxyl ester lipase, also known as cholesterol esterase, will then break down the monoacylglycerol into free fatty acid. So this will come out as free fatty acid, as well as glycerol. So this will come out as the water-soluble glycerol. There is an anti-obesity drug that is called Orlistat, and this drug covalently binds serine, the amino acid serine, at the active site of gastric as well as pancreatic lipase, and it inhibits this enzyme. And if this enzyme is inhibited, then the triglycerides will not be digested and absorbed. The phospholipid in this droplet will be acted on by an enzyme called pancreatic phospholipase A2. This enzyme prefers position 2, so this part of the phospholipid, and we get free fatty acid as well as lysophospholipid. So this would be your lysophospholipid. The cholesterol ester that is present in the lipid droplet in the small intestine will be acted on by carboxyl ester lipase that we talk about, your cholesterol esterase, and that will produce free fatty acid. So this fatty acid part is removed and you get cholesterol. The lipid droplets also contain fat-soluble vitamins A, D, E, K. And if any of these fat-soluble vitamins exist in the ester form, if there is any fatty acid attached here or here or here, then we have the enzymes pancreatic lipase as well as your carboxyl ester lipases. And these lipases can break down the vitamin esters into the free vitamin as well as your fatty acids. So as this digestion happens, we see increasing amounts of surfactants of the amphiphilic lipids such as lysophospholipid. We see increasing amounts of cholesterol that has a little polar hydrophilic group here. We see some monoacylglycerol that is a very nice detergent, surfactant, and then we see a lot of free fatty acids on the lipid droplet. So the increasing amounts of surfactant in this lipid droplet is going to make the droplet smaller and smaller, and in the presence of bile salts as well as peristalsis, what we eventually get is some very small particles that we call micelles, and these are mixed micelles, about 5 nanometer in size. So these are not micelles of pure bile salts, they are mixed micelles, so they are basically micelles containing bile salts and several other surfactants. So now we have this mixed micelles, very small mixed micelle particles that have a variety of polar lipids, surfactants, including sphingomyelin. So in the next step, there are two enzymes, alkaline sphingomyelinase, that's produced by the epithelial cells that line the bile ducts, as well as by the intestinal cells. And there's another enzyme, neutral ceramidase, that is produced by the intestinal cells. Sphingomyelinase will now remove the head group from sphingomyelin, and that will give us ceramide. The ceramidase enzyme will remove the fatty acid part. So this fatty acid is removed, and that gives us the free fatty acid as well as the product sphingosine. So at this stage, we now have these very small mixed micelles, and these mixed micelles will have the fat-soluble vitamins A, D, E, K within them, and on the surface will be all the lipids that have polar groups on it. For example, sphingosine, you can see the part that likes water, cholesterol, free cholesterol that has the small hydroxyl group. There might be a little bit of monoacylglycerol that was not completely digested, and you can see the nice head group here. A lot of fatty acids with the carboxyl, ionized carboxyl on the surface, the detergent bile salts, as well as lysophospholipids containing the head groups on the surface. So this is what we will have now close to the enterocytes, these mixed micelles containing all of these lipids. So these mixed micelles will now start accumulating around the glycocalyx. So just close to a glycocalyx, there is a water layer that is attached to the glycocalyx. This is an unstirred water layer, so there is not much movement around here. And so they'll be very close to the intestinal cells. Now this area here is acidic. This glycocalyx area, as we mentioned in an earlier video, is acidic. And that acidic pH is going to protonate the carboxyl groups of the bile salts as well as the carboxyl groups of the free fatty acids. So with the protonation of these groups, they become weaker detergents, and the micelle will now dissociate. Okay, so the particle will dissociate, release all the lipids, and the lipids will now be absorbed into the enterocytes. Okay, so that would be the next step. So let's look at the second step. So the first step was digestion. Now the second step would be absorption or the uptake by the enterocytes of the intestine. So the dissociation of the micelles will release bile salts, a variety of free fatty acids, including short chain, medium chain, long chain, a little bit of monoacylglycerol, glycerol, the lysophospholipids, sphingosines, the non-esterified or the free form of the vitamins, as well as the non-esterified form of cholesterol or free cholesterol. So let's look at how these lipids are absorbed into the enterocyte. Bile salts, remember, come from the liver. They are made by the liver, and then they are stored in the gallbladder, and then they're secreted into the intestine after a meal. Most of the bile salts, more than 90%, are transported or taken up into the enterocytes by sodium-dependent bile salt co-transporters. So from the enterocytes, these bile salts are sent into the portal vein, and from the portal vein, they will enter back into the liver. So basically, you can see that we have this enterohepatic circulation. Okay, so bile salts are secreted by the liver, and most of them are reabsorbed and sent back to the liver by the enterohepatic circulation. The short-chain fatty acids are believed to simply enter the enterocytes by passive diffusion, and then from there enter the portal vein, and the short-chain fatty acids are exported to the liver. The medium and long-chain fatty acids bind to the fatty acid transport protein, FATP, and then once they are bound, they're believed to diffuse to the plasma membrane and enter the cytoplasm. And in the cytoplasm, they will be bound by proteins such as fatty acid binding protein. Coenzyme A is then attached to these fatty acids, and they are converted to diacylglycerol, and then eventually to triacylglycerol, or the diacylglycerol can be converted into phospholipids. The phospholipids and the triacylglycerol can then either be stored as lipid droplets in the enterocytes, or they can be converted into chylomicrons, which will be then sent out into the lacteals. The lacteals are the lymphatic vessels of small intestine. Any monoacylglycerol will be taken up by fatty acid transport proteins. They'll bind to fatty acid transport proteins and probably then diffuse across the plasma membrane and enter the cytoplasm. And in the cytoplasm, the monoacylglycerols are bound by the protein retinol binding protein 2, or RBP2. Then they are converted to diacylglycerol, and as before, the diacylglycerol can become either phospholipids or triacylglycerol, and these phospholipids and triglycerides can either be stored as lipid droplets or exported out of the enterocyte as chylomicrons. The water-soluble glycerol will be transported into the cytoplasm by some transport protein and then combined with the fatty acyl-CoA to form diacylglycerols and then triacylglycerols. And these triacylglycerols and phospholipids once again could either be stored as lipid droplets or converted to chylomicrons for export into the lacteal vessels. The lysophospholipids are taken up by fatty acid transporters and once in the cytoplasm are converted to phospholipids. And then these phospholipids could either be part of lipid droplets or chylomicrons. Sphingosine is taken up either by diffusion or some sort of transport protein, and once in the cytoplasm is converted either to the fatty acid palmitic acid as well as the lipid ceramide. The fat-soluble vitamins A, D, E, K enter either by diffusion or by some transport protein, and once they enter the cytoplasm, they are packaged into chylomicrons, and these chylomicrons are sent out into the lacteal vessels. Now, cholesterol that may come from the diet as well as from bile, cholesterol as well as the non-cholesterol sterols, such as the plant sterols, they are transported into the enterocytes by proteins such as this one here called Niemann-Pick C1-like protein, or NPC1L1. And from there, they will combine with the the vesicle formation proteins such as clathrin and AP2 and form vesicles. So this cholesterol complex to the MPC1L1 will be in the vesicles, and the cholesterol then will be esterified. Fatty acid will be attached to it to form cholesterol ester, and this is done by the enzyme acyl coenzyme cholesterol acyl transferase, or ACAT, and this happens in the smooth endoplasmic reticulum. The cholesterol ester is then packaged into the chylomicrons, which are then sent out into the lacteal vessels. Now the drug azitimib binds NPC1L1, and this prevents the association of NPC1L1 with the vesicle formation proteins, clathrin and AP2, and thereby preventing cholesterol absorption by endocytosis. So by blocking cholesterol absorption from the intestine, this drug is useful for treating patients who have high blood cholesterol. Now it's important to note that cholesterol is not only absorbed by enterocytes, but cholesterol can be removed from enterocytes back into the cavity of the small intestine, and this is done by a process called Trans-intestinal cholesterol efflux, or TICE. So the protein ATP binding cassette G5G8, ABC G5G8, this protein here can actually remove cholesterol from the cytoplasm and send it back into the cavity for subsequent excretion into the feces. So this means that the amount of sterols that is absorbed into our body is regulated at the intestine. How much is taken in and how much is exported out or removed out back into the intestinal cavity. So in the disease, the rare genetic disease called cytosiderurgia, which is quite often misdiagnosed as familial hypercholesterolemia, these patients have defective ABC G5G8. So this protein here that is involved in TICE, this protein is defective, and therefore the excess cholesterol cannot be sent out. This leads to increased absorption of plant steroids and cholesterol, and these cholesterol sterol chemicals, they deposit in various tissues, forming what we call xanthomas. So in a previous video on cholesterol synthesis, we saw how the drugs called statins could inhibit cholesterol synthesis by inhibiting the enzyme HMG-CoA reductase that control cholesterol synthesis. And now we see another example of a drug that can actually inhibit cholesterol absorption, thereby lowering our blood cholesterol levels. So we can lower blood cholesterol by preventing its absorption, and we can lower blood cholesterol levels by decreasing its synthesis. Okay, so in the next video, we shall look at these lipoprotein particles. Okay, so we'll see what happens to these chylomicrons that are produced by the enterocytes, and we'll also talk about the other types of lipoproteins besides chylomicrons. So once again, thank you very much for watching this video and supporting this channel. Until the next video, have a wonderful day.